Hey, good afternoon. So today our speaker is Dr. Qiqi Deng from Moderna. Qiqi is a director in Clinical Biostatistics. She leads a group focusing on clinical development of public health and latent virus vaccines, as well as translational medicine research and statistical research. Qiqi has extensive experience in pharmaceutical industry. Before joining Moderna, she works for 13 years at Boehringer Ingelheim, with 7 years serving as leading statisticians for multiple projects across different clinical development phases and therapeutic areas. After that, she worked as a methodology statistician for 6 years. Her research interest includes hypothesis testing and modeling in dose finding, adaptive design, and pragmatic considerations in applying innovative methodology into real clinical studies. But without further ado, let's welcome Qiqi.
Thank you very much, Zhihua, for the introduction. Can you hear me okay? Okay, good. As Zhihua mentioned, today I'm going to give you a brief introduction for drug development and also how biostatistician can contribute. Just a heads up that the talk will be very non-technical. So it's going to be a high-level overview. If you are someone who already working in the industry as a biostatistician, you may find this talk maybe kind of a fundamental and basic. But I think the idea for this presentation is really to give people an overview, especially for those who are not so familiar with the drug development and how statistician can play a role in this process. To just start the conversation, this is a diagram for a typical drug development process. Everything starts from discovery, especially in the lab. And once a compound is created, then it needs to go through a series of clinical trials for testing. This diagram shows the attrition rate in each of the process. Basically, at the beginning, you may have identified 10,000 compounds. And then after the discovery phase, you're going to leave out a lot of ideas and proposals that's not going to work. And then when it comes to preclinical testing, you probably only have 250 out of that 10,000. And then among them, only a small number of them can move into actual clinical testing. And you can see that once you get to phase 1, phase 2, phase 3, and the phase 4 of the clinical development, it's really gradually you're having smaller number of compounds left in the pipeline. Therefore, this is a process with really high attrition rate. At the same time, the cost for the development becomes higher and higher if you move into the late phase. When you are starting the phase 1 clinical trials, it's approximately 20 to 100 healthy volunteers. And then when you go to phase 2, it's probably 100 to 500. And then for phase 3, it's typically 1,000 to 5,000 and even more than that. So as you can see, when you go deeper into the cycle, then the more sample size is needed and also the higher the cost. Therefore, I think in this process, you really want to control the risk very well, not only for the investment of the drug companies, but also for the benefit of the patient that they're getting the more promising drug. Okay, and then this slide summarizes the typical phases of a clinical trial. When we talk about phase 1, the purpose of the phase 1 study is really to evaluate the safety of the drug and also to have better idea about the dosage that we're going to use for subsequent clinical development. And it also helps you to refine your phase 2 design. and among this stage, only 17% of the drug, among this stage, actually 17% of the drug moved to the next phase. And then when you get to phase 2, then you start to test the drug in actual target population. The patient who have the disease start to take the drug. Then you start to observe efficacy of the drug and also the safety, the side effect. However, that really helps you to find out whether the drug works or not and help you to refine the research question to design your phase 3 studies. Among this stage, about 33% move into phase 3. Finally, when you come to the phase 3 study, this is usually the large scale long-term trial start. And we use this phase 3 study to confirm the drug is efficacious and safe, and only about 25% to 30% of the drug can move into approval. I'm sorry, I need to mute myself. And then after the drug got approved, you move into phase 4 or post-marketing studies. That's not going to be the focus of our conversation today. And then talking about how statistician can contribute within this process, this is a typical pool I used. I don't know whether we have the functionality to do that here, but you can think about if you're a statistician working in the pharma industry as a biostatistical lead, then what are the times you're going to spend on each of the perspective? Whether you're designing the study, you're oversighting the conducting of the study, or you're doing analysis. So between these top 2 options, the 2 roles I provided, what would be your guess in terms of how much time I allocate to each of the aspects? We're not going to do an actual poll here, but I'm sure you probably have an answer in your mind. Honestly speaking, when I typically have a conversation with someone who are not familiar with what we're doing in this industry, they tend to think a majority of our time are spent on data analysis. So some people even call biostatistician a data analyst. This is actually not true in pharmaceutical company or biotech companies. Actually, our time allocation is more like the first row here in the table. We spend quite some time in designing a study, and then we need to make sure the overall conduct is with high quality. And then finally, we conduct the data analysis. Although we do spend a lot of time on the data analysis, but the design and the conduct are equally critical. And once you move up in terms of having a larger responsibility, oversight, more and more projects, then your focus and your time will be more heavily spent on the design of the study instead of the actual data analysis. So I think across my talk, you're going to see that I have little information on how you analyze the data. I don't spend too much time talk about the actual data analysis examples or results. A lot of the focus will be given to the design of the clinical trial, and that is actually in the core of our job function. So this is what I mean for designing clinical trial. Usually when we start a clinical trial, the first step is to design it. A typical clinical trial is trying to answer a very specific research question. And then the trial protocol is a study plan that tells you what to do to run this whole study. It provides you a lot of information, like who are qualified to participate, which is inclusion exclusion criteria, how many people will be part of the study, how long the study will last, and whether there will be a control group like a placebo or active drug, and then whether there are other ways to limit the research bias. Then how the drug will be given to the patient and at what doses. What are the assessments you're going to collect, and how the data will be reviewed and analyzed. So the protocol really covers a wide range of the topics. We're not the lead for all these topics, but for two of the topics, the first one, how many people will be part of the study, and secondly, how the data will be reviewed and analyzed. I think it's easy to imagine that biostatisticians are the leading roles for those two topics. However, in order to have a great design for a protocol, as a biostatistician, you need to be familiar with all the questions that's being asked here in the list and also beyond the list. Okay, so then that's overall hopefully setting the stage of the talk. And next, we're going to dive a little bit more into each of the phases in the clinical development and see what are the relevant clinical questions and statistical questions. So we start with phase 1 study. Phase 1 study is usually a dose escalation study. And the primary objective for that, first of all, is to estimate what is a maximum tolerable dose. Or sometimes we want to find the maximum feasible dose. If a tablet is too big for you to swallow, and you already have 5 tablets, you don't want more, then you don't want to go more than that. So there are maximum tolerable dose in terms of the adverse events, and then there are maximum feasible dose. But regardless which one we are talking about, we're really talking about finding the upper limit of the dose that you want to use in the clinical trial. And then secondly, if a compound is very safe, even if you go up really high, you didn't observe much toxicity, then you probably want to use related pharmacodynamic markers to guide you to find a dose that is active to your body pharmacologically. For this dose, we call it pharmacological active dose. And then for oncology, we usually use phase 1 design to define the recommended phase 2 dose. We're going to cover the oncology in a few separate slides once we talk about the typical phase 1 and phase 2 studies. And to summarize that process in phase 1 dose escalation study, you can imagine when you have a new drug to be tested, then the first thing you're concerned about is the toxicity, whether it's safe to take the drug. The usual way we do that is we start with a very low dose, and then we gradually escalate the dose to make sure everybody is safe. So from low to end, that's a dose escalation. From the low to high dose, that's an escalation for phase 1. The dose we started with is usually called maximum recommended starting dose. And I'm going to talk about how to determine that in a minute. And then we escalate until we feel this dose is too toxic. We don't want to move up anymore, and then we call that maximum tolerable dose. In terms of how to select the starting dose, what we do is we usually have the preclinical toxicology information, and we have pharmacology data and many other data. And then based on that, especially the animal studies, what dose is considered safe and tolerable for the animals, then we are going to do a little bit translation and convert it based on the human equivalent dose. After we get that human equivalent dose, we're going to apply a safety factor, maybe take 1/10 or 1/4 of the human equivalent dose to make sure that the starting dose is safe. And then we use that as a start point for this study. So there are a few words that's very commonly used in this process. And if you're working in this industry, there are so many acronyms that you need to get used to when you start your work, including but not limited for those listed here. I talk about beside the safety, sometimes we can look at the biomarkers or pharmacodynamic markers to understand whether the dose is doing something and pharmacologically active. And it is okay to use that prior knowledge to guide your dose selection. However, there are many caveats for using that. First of all, in order to know what dose might be useful, then you need to have a very good understanding of the animal model of the disease and the mechanism of the action. And that knowledge is not always available. And then the information about the target site or receptors may be absent or modified. So in some of the cases, you probably are not going to get a lot of information before you really start the clinical testing. And then for some other cases, even if you have the prior information and you have the extrapolation, it may not be that reliable because the extrapolation from animal to human can be off. And then the route of administration are often different between preclinical and clinical testing. And also the pharmacodynamic effect could be different from the clinical benefit. The last item, the pharmacodynamic effect versus clinical benefit, it could be summarized by the following graph that when you increase the dose, you should be expecting a better pharmacodynamic effect in terms of the efficacy. However, the clinical effect may follow a slightly different curve, and it may take more doses to reach the clinical benefit. And for some of the dose level, even if the pharmacodynamic effect is zero and we see a change in the biomarker, but it doesn't mean the real patient will actually see a change in their disease and to feel that clinical benefit brought by the drug. So there are some differences between preclinical and also early clinical biomarkers versus a final clinical benefit. For phase 1 study, if we're talking about chronic disease, and generally in the non-oncology field, we usually use healthy volunteers for phase 1 testing. The study design is more or less like this, that you sort of have different groups lined up. For the first group, for example, you can assign 3 subjects on the testing drug low dose, and then 1 subject on the placebo. And then once you confirm this group, this dose is safe, you move on to group 2 and try a higher dose. And you just keep doing that. For the first study, it's usually called a single rising dose study, that you just dose the subject once and then stop there. Once you get that single dosing information, then you start the multiple rising dose study that you give maybe the subject 30 days and take one tablet each day and do that for 30 days. And then that's called multiple rising dose study. You also follow the similar escalation scheme under the multiple rising dose study design. And then in terms of the final analysis, you can compare the different dose groups across. And also you can combine the placebo patient across different groups too for your control arm information. So those are the phase 1 study design. And then once you find that upper limit of the dose that you don't want to go higher, that maximum tolerable dose, then that's where you sort of wrap up the phase 1 testing. There are many other things you want to do in phase 1. For example, establish the pharmacodynamic profile of your drug and establish more understanding about the biomarker and clinical benefit that might be coming there. There are a lot of things you can do in phase 1 study, but I just want to emphasize the two fundamental things you want to do. First of all, is to find the upper limit of your dose range, and then secondly, to understand better the pharmacokinetics profile. Okay. So I will pause here a little bit, and then we can move on to phase 2 study. For phase 2 study, the research question is going to be changed from phase 1. For phase 2, there are two primary objectives. The first one is you want to establish the proof of concept. And those studies are also called the phase 2A study. What I mean by establishing proof of concept is that you just want to have an initial signal that whether the drug works or not. If the drug works and deserves further investigation, then you can move on. If there is no signal at all that the drug is working, or the effect is so small you don't want to have continued investment, then you stop there. So this is called a proof of concept. In the proof of concept study, you usually use the highest dose against the placebo, the highest upper limit that you find out in your phase 1 study. The reason we do that is because for the proof of concept, we want to maximize the drug effect so that we give them the maximum chance to succeed in a proof of concept study. And usually there is a monotonic assumption that if your dose is higher, then the drug efficacy is better. So that's why we typically use the highest dose against the placebo for proof of concept study. And then the second objective is to do further dose finding and find out what is the recommended dose that you're going to use for the large study. For the large study, you really want a dose that have a better chance for a good benefit and risk profile. Therefore, you have to do a little bit more exploration in your phase 2 to find the right dose. Such kind of dose, phase 2 dose finding study usually have more than 3 doses. This is not the case for oncology, and again, we're going to talk about it separately. And then we are also going to keep looking at efficacy data and safety data. The typical question we're trying to answer in the phase 2 study is basically as simple as this, that you have a null hypothesis. The test drug is similar to placebo, it doesn't work. And then you have alternative hypothesis, the test drug is better than placebo. When we use the mathematical terms to reflect that, it's basically the mean of your primary endpoint. Let's see if you're going to lower the blood pressure, then that's the mean of the blood pressure values. And then you're trying to see whether it belongs to non or alternative. And for people here as a biostatistician or statistician, you must know R.A. Fisher, and he is a father of the modern statistical science and experiment design. And then he created the p-value. And just want to remind everybody that when Fisher created the p-value, it's supposed to be a continuous value, and it's sort of an index to measure the discrepancy between your non-hypothesis and the data. And then when it moved to drug development, you oftentimes talk about this magic 0.05% of type 1 error or alpha you're using in your experiment. This is based on the Pearson and Neyman decision-making framework that if you have a true hypothesis, whether the drug works or not, then you have the results of the experiment, then you can reject or fail to reject the non-hypothesis. And that lead to type 1 error or type 2 error. So I'm not going to do the stats 101 here today. What I want to emphasize is when it comes to drug development, as I mentioned earlier, the non-hypothesis is a test drug is similar to placebo. So the difference between the two are close to 0. And then your alternative is the difference between the two is, for example, larger than 1. And then you really want to find the cutoff of the data to tells you whether the drug works or not, then you can move on to the next stage. And then correspondingly, there are type 1 error and type 2 error associated with that. And you can calculate the sample size based on alpha, beta, treatment effect size, and also the variabilities. So this is your typical stats question. And then when you have only a high dose versus placebo, it's a two sample test problem. However, if you want to combine the proof of concept with a dose ranging study, and you are having multiple doses in your clinical trial, for example, you may have 4 doses, low dose, medium, 3 doses, low dose, medium dose, and high dose, and then you have the placebo arm, and you randomize the subject into the 4 arms, then how do you do the testing under this multiple dosing scenario? So now if we think about the dose response relationship, when you increase the dose, there could be very different types of dose response curves in terms of the response for efficacy. And you could have very little activities at the lower dose range, and then the high dose works well. Or actually from a very small dose, you start to see a strong efficacy effect. So there could be various type of dose response curve. And then how do you prove your drug works under different assumptions? We know that there is a donut test for pairs comparison to placebo. And then you could make the assumption that there is a monotonic going on when it comes to dose response, and then you can just use a highest dose against the placebo. Or you can combine the data in different ways and give different arms the different weights. However, if you do that, it needs to be robust. And the two of the robust ways to do that are trend test or the model-based contract, multiple listing adjusted model-based contract method. So basically, this is a quick illustration for the trend test. In this idea, you sort of combine the different doses using a certain coefficient or contrast for the testing. For example, if you have 3 doses, then you use -3 times a placebo, -1 times a low dose, then 1 times a middle dose, and 3 times a high dose, and you average, you sort of add them up. So essentially, you're comparing the higher dose versus lower dose, but giving the highest dose and the placebo the maximum weight at the two ends. So basically, this kind of testing are using ranks of different treatment groups to do to come up with a contrast test. And then there is a MCP mod approach where you can start with different possible candidate shapes. And then for each of the shapes, you can come up with optimal in terms of most powerful test. And then you can apply all the tests and find out which one is successful. And as long as you have at least one success, then the drug works. So as a biostatistician, you are going to get yourself familiar with the different designs and the analysis approach when it comes to this kind of clinical trials. This table illustrates how much the statistical model, how much difference the statistical model can bring in on the table. For example, if you're using a simple buffering adjustment, the total sample size could be as high as 390. And if you're using the trend test or MCP mod, then the sample size is about 230 plus minus. And just to keep in mind, those different approaches achieve the same power for the testing. But by utilizing the more advanced approach, actually, you can achieve the same power with a much more reduced sample size. So that's the proof of concept part to demonstrate the drug is potentially working. And then the other thing we mentioned for phase 2 is to find out what dose is the best for later testing. For that, I think we do a slightly different process that we go from the higher dose to the lower dose. We go backward. The reason is that although we know the maximum tolerable dose probably have the best efficacy, but maybe the adverse events associated with that is too strong and we want to find a better dose, then potentially we can find a dose that still have reasonable efficacy, but with a much better improved toxicity profile. So that's a purpose. I think I'm going to skip some of the technical details about this design, but there are a very important concept called minimum effective dose, which is the smallest dose with a discernible useful effect. And then by trying to find that minimum effective dose, we usually need to test on some subtherapeutic dose, on which the response is clearly below the maximum effect. So if you have a dose that you expect to be working, then you're going to test it. Sometimes there is a hesitation to use a low dose because of the fear that the dose doesn't work. But for a good drug, actually, it's very typical. Even a small dose can work pretty well. And then if you don't go to the lower end, actually, there is a harm to that. This is one of the examples in a phase 3 study conducted by Pfizer many years ago, where they explored a few doses in the first study, and then find out all the doses are similar, and then they try to add another dose, and then find out in the second study that it's still similar, and then finally it takes 3 studies to find out the real minimal effective dose. So the impact on this repeated dose finding is going to be very big on the project timeline. For example, if you start a phase 1 study in 2008, and then phase 2 study in 2009, and then it takes 1.5 years to do one study. And if you keep doing that, you actually could delay the development by a few years, and subsequently delay the drug to the market and to the patient to help them by a few years. So this is going to be a big thing for either the drug company or the patient. And if we look into the literature, there are a lot of information about what are the possible reasons for the delay or denying of the FDA approval of the new drug. So among those different reasons, actually the uncertainties related to dose selection is definitely the biggest. And then if you look at the drugs that already reached the market and then have a post-marketing dosage changes, this number is actually pretty high. About 1 in 5 new molecular entity drugs will have a post-marketing dose change. So that means the dose optimization or dose finding in the early phase is very challenged, and there are a lot of work we can do about it. So there are a lot of learnings from that, and there are workshops, and there are recommendations from different parties. Actually, in the end, I think some of the most useful rule of thumb I find is that, first of all, you should include at least 1 dose at expected subtherapeutic level to inform your dose selection. And then secondly, it's useful to come up with a wide dose range for testing. And then I will skip this one and move on to the oncology development. So for oncology development, I mentioned earlier, the oncology drug or cancer treatment will have different process for development. This diagram summarizes the difference between the two. For chronic disease, that's where we spend the last 40 minutes or so talking about. And then when it comes to oncology, you can see that it still have this proof of concept study phase. And then it also have the registration confirmatory phase 3. However, the main difference between that is how the dose is being selected. For oncology drug, most of the dose selection happens in phase 1. At least that was a history. However, nowadays it's start to change. When we talk about the phase 1 study, the difference is that the oncology treatment are usually very toxic in the old days, like the chemotherapy, the first generation of the chemotherapy. So for those cytotoxic agents, there is a lot of evidence for monotonic assumption for efficacy. And then the phase 1 dose escalation is trying to really push the dose to be as high as possible. In this way, it can really kill the tumor So the study design is, the study endpoint is usually based on those limiting toxicity, and those are higher grade, like grade 3 or grade 4 events. And a typical rates we're targeting for it is to find a dose that close to 20 to 30% of the subject will have those limiting toxicity. So we don't want to go too low because the toxicity and efficacy usually comes hand in hand in the traditional chemotherapy. And then because of the life-threatening nature of the disease, if you have the starting dose to be too low, or if your escalation is too slow, then that's going to be a concern, because then the subject is not likely to be treated adequately unless you already put them on standard of care. So therefore, the whole dose escalation design is different in oncology. They usually start with a marine LD10 dose. That means this is a dose that will lead to 10% of the mice to be mortal. So that's a mortality rate of 10% in the mice. And then you translate that to the human equivalent dose, and then you sort of take a safety factor of 1/10 or so, and use that as a starting dose. And then when you do the escalation, usually at the beginning, you escalate a little bit more aggressive, but when you reach a higher dose, you're going to become more and more conservative. Therefore, the modified Fibonacci is usually in terms of defining the dose sequencing. And there are a lot of statistical designs help with that. Traditionally, there are 3+3, and then later on, there are parametric models or hybrid models. So the different models really help you to increase the chance of finding the right dose. This is an illustration for a simple 3+3 design that you just dose 3 patients on the low dose. And if all of them are safe, then you increase the dose. You increase the dose until you observe the 1 out of 3 DLTs among the subject. Then in that case, you add another 3. That's where 3+3 design comes from. And then if you have 1 out of 6, you can keep escalating. But if you have 2 out of 3, then you're going to stop there because that's considered too toxic. So that's a very simple 3+3 design. And nowadays, there are a lot of models that's much more sophisticated than 3+3. They're Bayesian-based approach. They're rule-based approach that's fundamentally comes from Bayesian method. And there are many, there are model-based approach trying to model the whole dose response curve. So overall, I think the dose response design is very different in oncology. But oncology treatment also have many other special characteristics. For example, in oncology, a cancer treatment can potentially work for multiple cancer types. One treatment can work for colon cancer as well as breast cancer or head and neck cancer. So there could be multiple indications for one drug. And then there are a design called basket design handling that. And then for oncology, it's also typical that nowadays, based on the biomarker profiling of a subject, you could pick the right treatment for them. And we call that personalized medicine. And in some of the studies, you can bring in different treatment options. And then based on the screening results from a subject, then you can find out which treatment they're going to receive. And that's called umbrella design. And then in terms of the oncology endpoints, there are tumor response endpoint based on image data. There are progression-free survival or overall survival data, which is time to events. And typically, if a subject have a progression from cancer, then you're going to switch them on other treatment. That treatment switch or treatment crossover cause the complications in analysis. And then if you observe some very, very promising tumor response data or early endpoint data, are you going to make this available for a subject who are waiting for a treatment? So then that comes to the question whether the endpoint have the surrogacy relationship with the final clinical endpoint. Therefore, in summary, oncology development have its special challenges, but also a lot of opportunities for statisticians to work on. Okay. Then finally, I think we're coming to the phase 3 confirmatory study. A phase 3 confirmatory study is a basis for the registration. It's very typical to require 2 positive trials. However, for rare disease, fatal disease with unmet need, the requirement may be different. And those studies are usually long-term, large-scale study to evaluate both efficacy and safety of the drug or treatment. And then finally, the sample size is usually determined based on efficacy testing as well as minimal safety database requirement. The first one is more a statistical question, but the second one is not necessarily a statistical testing question. For us, one of the biggest difference I find between my years at school versus my current work is that when I was working in school, I think we usually do the analysis after we receive the data, and we can try different method on the data set and find out which method works best. We don't have that level of luxury in clinical testing. It's very important to pre-specify the primary analysis. The reason we're doing that is we want to minimize the cherry picking effect and the associated bias. And then whatever model we choose, it needs to be robust. And we want to make sure whatever we see in the end, if we have a difference between the test drug versus placebo, then that difference is caused by the treatment, not by something else. Therefore, maintain that causal inference is the key. So we have to go back to the Pearson and Neyman hypothesis testing framework again for decision making. And this again is the same statistical question you're trying to answer. However, between phase 2 and phase 3, type 1 error and type 2 error have different consequences. In phase 2, if you have a type 1 error, which means it's an ineffective drug, but you move it to the next stage, then the consequence is that you just move an ineffective drug to phase 3. And when it comes to phase 3 study, the same type 1 error concept. However, the consequence is that you will have approval of an ineffective drug for wide public use. Therefore, this level of consequence are different. That's why the agencies are very, very serious about the type 1 error control, because they don't want to approve a drug for public use if it is not working. And then the power or the type 2 error beta also have different consequences between phase 2 and 3. For phase 2, the type 2 error means that you're giving up an effective drug. And then for phase 3, it means that the effective drug cannot be approved. So the phase 2 is more, it sounds more like it's relevant more for the drug developers. However, I think either way, it is a loss for the public health field if you're giving up an effective drug. So those are the implications for the statistical terms under the clinical testing setting. And then finally, I'm going to briefly touch upon a couple of important concepts or topics that biostatistician will be heavily involved in the discussion. The first one is called intention to treat, or the ITT principle. Again, this is not a concept that we learn in school in a regular basis, unless you have a special class designed for clinical trials. However, this is a very critical concept for statistical principles in clinical trial analysis. I copied the definition or the explanation from ICH E9, and in the high level, it means that intention to treat is, if you have an intention to treat this subject, you're going to include them in the analysis, and you're going to use the initial randomized arm for analysis. But there is a much broader meaning for that intention to treat, especially when it comes to missing data or the intercurring events handling during the clinical study. For example, if a subject discontinued the treatment 3 days or 1 month after the initiation of the treatment, then are you going to use their data or not? That's a very typical question we're going to face in a clinical development program. There are all kinds of reasons or intercurring events that will cause problems in analysis. So like I mentioned earlier, there is could be a treatment discontinuation, and there could be a rescue medication use if the subject is not doing well. And then there could be treatment switch, there could be death, there could be a treatment discontinuation due to lack of efficacy. So all these intercurring events will lead to similar consequences that you're not going to be able to observe what you want to observe for the primary endpoint. For example, if we still go back to the blood pressure lowering drug that I mentioned at the beginning, if your targeted endpoint is the blood pressure 24 weeks after treatment, and then you just have an intercurrent events before that, and you drop out from the study, for example, then we're not going to have your data available for that 25-week time points. Then how are we going to do with it? A simple completer analysis would say that if I don't have the data, I don't include the data. I just ignore that patient. But this is really problematic because if a subject discontinue the treatment due to adverse events, that means this drug is really not working for this subject, right? They cannot tolerate the drug. Then this is a very important piece of information you want to consider. This is a treatment failure in a lot of cases. But if you just throw away this patient data, then that leads to bias in evaluation of your treatment effect. Therefore, I think we need to be very careful trying to stick with intention to treat principle in a lot of cases. However, this question is probably more complicated than just the intention to treat principle. That's why I think in the past few years, actually, there is a ICH E9 addendum to talk about a framework to systematically handle the different type of intercurrent events. And I'm going to quickly go over that. For example, if you have a if you have a treatment discontinuation and you keep following up the subject and still collect their data for analysis, then that's a treatment policy, or it's following the original intention to treat principle. Or if you're stopping the treatment based on tolerability issue, then you can claim this is a treatment failure. Then we can call it a non-responder imputation. On the other hand, if you want to say that, what if I continue the treatment, then what will be the predicted or estimated response from this subject? Actually, some of the modeling approaches doing that. And then for a while ago, there was an idea that you just carry over the last observation from the subject to be the final response, and that's called last observation carry forward back then. And then you can consider it as a while on treatment policy. So there are a lot of terminologies. It may not make too much sense to you right now when you first heard about it, but when you go deeper into the topic, you'll find that these are a lot of wisdoms and sorts put into this. Finally, the last topic I want to spend a little bit time on is the multiplicity topic. This is another important topic for biostatistician in clinical development. So when we have a clinical trial, we usually have a primary endpoint, and then primary hypothesis that, for example, the drug reduce blood pressure at week 24. However, when it comes to actual study, it could be much more complicated than that single question. You could be testing 2 or 3 doses, and then you could be looking at different endpoints. For example, in oncology, you could be looking at tumor response, you could be looking at progression-free survival, you could be looking at overall survival. And then there could be subgroups. You're looking at females or males, older population versus younger population. And then you could be looking at different time points, like you're doing the analysis first look when you have 100 subjects, and then you're having a second look when you have 200 subjects. And also there could be a superiority claim that I'm trying to see whether the drug is better than the current standard of care, or there could be a non-inferiority claim that I'm trying to see whether my new treatment is comparable with the existing treatment. So there are actually a lot of potential hypotheses or potential questions you're trying to answer within a clinical study. And then if you just say, as long as I am successful for one of the dose, in one of the endpoint, in one of the subgroup, in one of the time point, then I have a successful drug, then you can see that the type 1 error can easily get inflated. One of the very intuitive simulation is that, for example, I have a placebo arm with 50 subjects, and each one with 30% response rate. That's what the blue curve shows you in terms of the histogram. And then if I consider I have 3 different doses, the low dose is just the same as placebo, which is indicated by the blue color. And then for the low dose and for the medium dose and high dose, actually it doesn't get better. It's still the same as placebo. So that means the drug simply doesn't work. Okay. Then if you look at all the 4 arms, it should be following similar distribution as indicated by blue color. However, if I start to cherry picking, let's say I look at 2 dose groups and pick the better one, regardless which one it is, I just pick the better one, then the distribution will shift to the right, like the red curve. And if I have 3 different doses and I again pick the best dose, then the distribution becomes the green color. So as you can see, once you add more and more possibilities for success, your distribution is actually shifting. That introduces a bias. And if you're just using the best out of the 3 doses as the test arm to be compared to the placebo, you're going to find out it's better than placebo, not because it's working, but because you're cherry picking. Okay, so that's why I think there are multiple works related to multiplicity adjustment. This is just one of the FDA guidance about multiple endpoint in clinical trials. It is one of the FDA guidance that have heavy content on statistics related topic. And then there is a concept called family-wise type of error that you really want to control a phase 3 study to have the family-wise type of error be below a certain level. And when we talk about family, we're talking about a family of hypotheses, and we want to make sure that the error rate is controlled for each one of them, regardless of whether for other hypotheses there is a benefit or not. So this error rate is very important in clinical trial statistical analysis. And there are different methods developed to control that. Just take a multiple sclerosis study as an example. If you have 2 dose levels of a drug with a control, and there are 3 endpoints, the annualized relapse rate, the number of lesions in the brain, and the disability of progression, then we can technically have a lot of different hypotheses. The high dose versus control, or the low dose versus control as indicated by I, or the 3 endpoints as indicated by J, then you can form technically 6 hypotheses by using different combinations. And then there are different ways statistician can offer to control the family-wise error rate. You can specify an order that you start from high dose to low dose for the first endpoint, and then do that for the second and the third. Then you can only go and test in the next hypothesis only if the previous hypotheses are successfully tested. Then we call that fixed sequence testing. And there is a simple Buffoni split that you just split them to be 6 portions, and each one got 1/6 of alpha. And then there are more complicated procedures that can be used to increase the performance, especially the power, but still get the type of error controlled in the appropriate level. And this is where really the statistician can contribute quite a bit. And the biostatistician actually are the one leading the discussions and trying to compile different input from the clinical side or from the marketing side, and then try to build a strategy that works best considering those aspects. Then I also include a graphic approach. this again specify the order of the hypothesis testing, and also how the alpha could be split in different hypotheses. So this is probably not really a topic for a 1-hour presentation, but just to have a quick demonstration how complicated the problem can get into when you have multiple doses and multiple endpoints. Okay, and then finally, there are two also big topics nowadays in terms of the innovations in the design. One is called adaptive design, which means that you're adaptively monitoring what happened within a study and then change your action about the study on the way. And you have to do that in the pre-specified manner in terms of all the adaptation rules need to be pre-specified. Therefore, we need to run a lot of simulations to evaluating the characteristics of such design. And then there are Bayesian analysis where you try to leverage the historical data into the new studies, or you're trying to combine the information from a lot of different resources to have a data synthesis activities. And then finally, I think for a biostatistician working in this industry, beside the statistical topics we just talked about, and beside the basic knowledge of the drug development, there are other things that will be very, very beneficial. First of all is the medical knowledge for the disease that you're working on. We're usually not by training a medical doctor. However, I think we have a lot of opportunities to interact with the doctors. So from there, and also from the experience of working on those trials, you actually naturally will gain a lot of medical knowledge, and that's going to benefit your contribution of the clinical development. And then for us, we always working a multi-discipline team. The drug development is such a complicated process that we really have to do it under a team setting. There need to be clinician, there need to be regulatory affair expert. There need to be someone who are expert for the pharmacokinetics or the biomarker. And then when you run a trial, there are a lot of operational aspect of it. And then you need someone to really closely look for any safety signals to ensure the patient safety is protected. And then there are market access from insurance or other point of view. So this is really a multi-discipline team for any project that you're working on, and the communication is critical under that setting. Okay, so I think that's the end for my presentation today. I understand this is a lot of information. I want to open up for any question you have, either from the content of the presentation or just generally about this industry. So I will hand it over back to Zhihua